button is here. And this should be live now. Working. Yes, great. Okay.
People are coming in.
I know, not everyone is on holidays yet. Let's just give it another minute so you don't get interrupted. Thanks for everyone who's joining us today already. This is more people than I thought it would be. This is nice. And we already have people over on YouTube. Just to explain the rules, how it works, there is a bunch of us live here on Zoom and a couple over on YouTube. The people over on YouTube are welcome to ask questions in the chat, and then I'll bring it over here and ask our speaker on your behalf. Now, welcome everyone. Let's make a start. Today we have Wietske from the Zwaag, who has a very interesting CV, an undergraduate in molecular sciences from the Netherlands, and then a PhD in physics and astronomy from the UK, Nottingham, where she developed pulse sequences for functional MRI, working on one of the first 70 scanners. And then that was followed by a postdoc in lovely Switzerland, also working on high-field fMRI. And since 2015, she's actually at the Spinoza Center for Neuroimaging in Amsterdam. And as I just learned, currently in the same city as I, in Nijmegen. Today's talk is going to be on mapping brain function using ultra-high field MRI and its clinical applications. And I don't want to take any further time away from you, so I'm handing over to you. Thank you for being here.
Well, thank you for the invitation, and thanks everyone for joining and not going to the beach instead. It's nice to be here. So yeah, as you can tell from my biography, the common theme is ultra-high field MRI for quite a while now. So when I put the talk together, I thought, oh, I don't usually do so much clinical stuff, so I tried to give it a swirl towards clinical applications, but mainly it's brain function and how and why do we use 70 to map this properly. Yeah, advancing slides, obviously. Lovely Zoom. Okay, so in an fMRI experiment, as you are probably aware, we acquire images, many, very fast. And while we do that, we ask our subjects to do a task, or we just do resting state and we look at spontaneous fluctuations, but over time always. So we have time courses that would look something like this, or like this, and then we say, well, we create a map of the parts that we find interesting and the parts that we don't, the networks that we care about, the networks that we don't. In all these cases, what we care about really are the fluctuations over time. So we're not looking at an image which is perfectly stable, but we're actually wanting to see time varying changes in the background of our MRI signal. So how can we improve then what we have? So if we look at the MR system, there are three main components: the static magnetic field, or the main magnet, the gradients, which are relatively independent. Even in an independent object, you just shove the gradient coil in and it'll work at any field strength, and the RF, which is tied closely to the field strength. Your pulses have to be at the right frequency for the nucleus that you're trying to measure. So what sort of magnets do we have then, if it's the magnet that we're going to change? Well, the common ones, and 1.5 and 3T are really the only common ones. They are everywhere. There are multiple ones inside the building near Stephanie's. So, and what we currently call ultra-high field is 70 and higher. Now, that's of course a bit inflation sensitive. So I think 20 years ago, 3T might have been ultra-high field, and in 10 years, maybe it's 10.5, maybe it's 11.7, but for now, 70 is ultra-high field. And that's also the system that I'm working on, so 70 Philips one. So why does that field strength matter? Well, a surprising number of variables change when you do MRI at higher field. There's pros and cons, or challenges, as you might want to call them. The first and most important one is the higher SNR. So if you have a stronger magnet, there's more signal. And really, that's why people pay so much money for having more expensive scanners also. But in addition, we see increased susceptibility-related contrast. And when you're trying to do fMRI experiments, that means there's more distortions throughout your images if you don't adapt your protocol. There's a longer T1, which can be important for many different applications, maybe less so in fMRI. There's also higher B1 in homogeneity and a shorter T2. And very similar to that change in susceptibility, also a shorter T2 star and an increased bold contrast. So if we look at this fairly long list, then these are probably the ones that are relevant for fMRI experiments, and whether those are clinical or not. These are things that you should at least consider before you set out making your protocol. So to go through these, if we look at the SNR first, then in any experiment for protons, there's 6 more spins aligned with the field than against per million at 1 Tesla, and this scales fairly linearly. So at 70, there's 42, which is still a very small number. So the signal we're looking at is still very noisy. And if you're used to fMRI experiments, you'd know that that's true. But it also means that there's lots to gain. Right? We can improve on the SNR for experiments in many different ways. So if we look at a comparison across field strength, then you see that it's a super linear increase, which is roughly anywhere the same anywhere in the brain, going here from 3 to 7 to 9.5. And when you look at the images, then of course it's the same brain, but in this case, you can really see that there's more speckly stuff at the 3T image than there is at the 7T and at the 9.4. And these are, again, same thing, similar coils. So you do nothing and you still gain. But you can also see that there's little contrast changes and that the images feel a little different, besides the change in SNR. So what we do usually with that SNR increase is that we trade it for higher spatial resolution. And now here I just realized that the timing of this figure is the wrong way around. But if you go from 1.6 millimeter, where you cannot really see the cerebellum, which is what I was trying to image here, to 0.8, and at 0.8, you start to see that there is actually a white matter tree, and then possibly you can distinguish some of the lobules. But really, you have proper definition of the anatomy at 0.4 millimeters. So this is something that you might really want to invest in a high field scanner to do. And that what we're trying to get to is this 0.2 millimeter scan. And here you can see that the noise is starting to get back at us. So here the voxels are too small to have decent SNR to have a good segmentation, for example. So higher spatial resolution. And we can do that in fMRI as well. So this is a very old image by now. It's a 0.8 millimeter isotropic EPI averaged over a few volumes. And then you see that there's nice definition of the anatomy. You can actually see all the draining veins. So you're not having so much trouble having to match this from an anatomical scan. You can take lots more information out of the functional data already. So high res is good. You could also use it for shorter scan times. And that can matter a lot if you're setting up things for learning or for patients that don't want to stay in the scanner for very long. Just being able to do the same thing but in shorter time, that can make all the difference to your experiment. Now, a big additional advantage of 7T is the fact that we're not just having higher SNR, we're also looking at changes which are much bigger. So this is a motor task, which a poor subject had to do with 3 different scanners at 1.5, a 3T, and a 7T. So it's the same ROI, same, well, as much as possible, the same bit of tissue, the same task, and the same amount of T2 star weighting, so the same amount of bold contrast. And then you see that the percent signal change from going from 1.5 to 3 to 7T increases almost linearly. So we have not only that SNR increase, but also just a much bigger thing that we're studying. So if I exaggerate that, no, I didn't even exaggerate this. If I simulate this, then I take a time course here where the SNR is, the noise is roughly as high as the bold signal. And in the case of the 3T, and then if I double both the SNR and the bold signal itself, then it jumps like to an extent that now, instead of having some noise where possibly you see a response, you can see that there is a response. You have a much more reliable, you know much better what's happening in the time courses. And that means you can go back and study things which are maybe a lot less obvious. So rather than doing a motor task, we can look at responses which are much more subtle. And the example that I picked here is from one of my colleagues who was looking at numerosity. Numerosity is important for survival, but not something that we're, if you want to have, say, 6 sandwiches or 2 sandwiches, it matters a lot to how many there are in front of you. But it's not something that we can easily map. They're relatively noisy stimuli. Now, in this case, these maps have been shown at 17. So we knew they exist. We knew roughly how the protocol should be set up to go about them. And we made the effort of going to 3T and just repeat the experiment until we saw that there were responses also at the 3T. And we found that it is possible to do these experiments on the 3T, but to achieve the same amount of explained variance, you have to scan for 4 to 5 times the number of sessions that you require at 7T. And that means that a lot of these experiments become infeasible. You can scan someone for half an hour, 45 minutes, maybe, but 5 times half an hour or 5 times 45 minutes becomes really too long, and it gets increasingly difficult to have volunteers agreeing to do this, but also to inflict this on the clinical population. So does 7T fMRI work out of the box? Well, not quite. I'm not sure how many of you have tried, but you will see that there are issues. So the most noticeable one is, of course, that there is loads more activation. And then if you're using the exact same analysis protocol, especially smoothing, you will find that that activation just goes everywhere, and then it stops being so informative. On the other hand, the EPIs, and that's why I took this figure here, become more distorted. So this is, again, that same motor task with another single subject. And now if you look at the background images, you can see that the EPIs get gradually more distorted as you go up in field strength. Still, that's helped by going to higher spatial resolutions, because then the distortions become relatively less important. So here, the high resolution is not just an advantage, it's also a way of, not a target, it's also a way of getting rid of the distortions that you didn't want in the first place. Another thing to keep in mind is the fact that you're trying to image bold signal changes or other functional changes, and not all the other things that are also happening, not the system drift, not the heartbeat, not the respiration, not the motion either. So once you're in a very high SNR situation, then your temporal SNR becomes dominated by all these things that are changing, but really you only care about a small part of those. So you might be getting a higher and higher image SNR, but not achieving much more temporal SNR because possibly your subject is breathing too much. In that case, you're not gaining as much from going to higher field strength. So then it'd be better to say, oh, I'm going to design an experiment where high spatial resolution is useful, or I want to scan very fast, or I have very difficult tasks that I need to repeat a few times, and then stay in this area where the temporal SNR actually increases linearly with the image SNR to have more benefit from the field strength that you're investing in. Here's a nice equation that describes this curve where the temporal SNR will follow the image SNR until it reaches that plateau, which is defined by Laminar. Another way of using the increased sensitivity and SNR is to invest in specificity. So we're mostly using gradient echo based bold in the entire field of fMRI, but you can choose to look not so much at T2 star changes as we do in the gradient echo, but at T2 changes, such as in the spin echo EPIs or in a Grace acquisition. And if you do that, you see that the responses are smaller, but they're also much less dominated by draining veins. So in this graph here. If you see my mouse, the peak is much earlier. So that we're looking at smaller venules, not so much at the draining veins. And as you saw also with the amplitudes in the time courses, these spin echoes are, the responses are theoretically half as big, and in practice sometimes even a bit smaller. So it costs, you take a big hit in the sensitivity. So on the one hand, you gain a lot by moving to higher intensity, by moving to higher field. So these sort of increases also make it possible to then use more specific acquisitions. And if you apply some time and effort and lots of patience and averaging, this can lead to really beautiful results. So this is a spin echo map of ocular dominance columns, one of the earlier applications from 70 from Minnesota, where the red and blue in the B panel are the inputs from the different eyes in V1. And then they look at the orientation sensitivity columns on the right. So these are feasible, but it's a very niche application. You have to, with dedication, you will get. Another very specific, spatially specific option is VASO, and we've been seeing a lot of these in the recent papers. This is one of our own data sets where we also have a VOLD and a VASO experiment, both for a motor task. And these CBV changes, so cerebral blood volume changes, are thought to be much more specific to the gray matter. And because it's a blood volume experiment, they said the blood is null, the signal's close to zero. And then if there's more blood, the signal gets lower still. So they're of opposite sign. If you look at the time courses, you see that the VOLD and the VASO will go in opposite directions in the plot below. It does follow the gray matter strip much more tightly than the VOLD, but you pay in the temporal resolution because you need to spend time doing that blood nulling. There's often smaller coverage to make sure that you can fit the readouts even in those 3 seconds. And the sensitivity is also not very high. So this will also work well at 70, not so much at lower fields. Then another, and this is really a disadvantage. I try to keep balance between advantages and disadvantages of the different changes, but same as an SNR increase is always good. B1 inhomogeneity is just tricky to deal with. So you see here in the background that there's more variation in the signal intensity. It's more difficult to achieve an homogeneous flip angle across the brain. And that happens because the wavelength is much shorter, and we can counter that by applying pulses on a multi-transmit system where we calibrate the pulses, much like a microwave is calibrated to heat your food throughout the microwave. We do something similar with the brains inside our scanner. So here I outlined the cerebellum because that's where I would like to have signal. And if I compare that to in the standard system on the left, and then if I compare that to the multi-transmit on the right, then there's lots more signal, and also the white matter looks more homogeneous. And you can apply the same approach to the functional data. So then there's clusters where you expect them. So that would be the technical challenges, moving to brain function mapping at high fields. The easiest maps, the most well-studied maps that we have in the human brain are those of the visual space around us. If we look at the literature, you often see them as occipital lobe maps or visual regions, let's say. And if we do the same approach at 70, and this is a huge data set, so they have massive SNRs, then you see that these maps really, they spread everywhere. There's maps of our visual space all throughout the brain, even in places like the default mode network. Given enough SNR, you will see these patterns over and over again throughout the brain. Then a different sort of map is a tonotopy, so mapping tones. And there's actually loads of work on this. I just took here some work of my own to be able to show you the example. So here we're mapping series of tones and then mapping the responses in the auditory regions, and you get this very nice high to low gradient over Hessel's gyrus with reasonable. So this is not sub-millimeter activation. It's reasonably high resolution, reasonably fast. So we can do this in a very short amount of time, which means that we could do it for individual subjects. And as part of the scan session, we can also add a manipulation of this map, which is what we did here. We asked people to attend to either high or low tones while playing them both. And you can think of this as attending to a conversation while you're at a cocktail party, which is what's called the cocktail party effect. So you can attend left, can attend right, very much on cue. And then you can see that the bolt responses for the given frequency will also follow the cues pretty nicely. And here it matters that that the initial mapping step is fast and that you can do it in individual subjects because the ROIs are relatively small. Something similar then applies in our semantic topic mapping, where we're mapping fingers of a hand. And we did this in a series of individuals, and we've done it many times since. And you see that there's nicely organized progressions from thumb to little finger and back over the sensory cortex. And again, it's all individual data. And then we can change what happens in those representations by asking people to do a task in the scanner. And here we opted for a rubber hand illusion, but an MR compatible version of the rubber hand illusion. It's difficult to do that in the scanner, where you ask people to stick out their hand and then you get them to stroke their finger and the hand that's placed against it. And then you see that the representation of especially the stroking hand is really enlarged, and as you would expect from what you feel when you're doing this. It's too bad we're on Zoom, otherwise I'd get you all to practice on your neighbors, but you can do this at home. Similar to the visual maps, there are body maps throughout your brain. There are double ones in the cerebellum, so we looked at those as well because they're even smaller, so we're curious whether we could manage to disentangle these maps. And we see these hand representations here, blue, green, yellow, red, orange for the 5 different fingers, also in the cerebellum. And then we expanded on this to see if we could see larger maps, and now we're trying to also manipulate what happens in these maps for different tasks. But those are still not the sub-millimeter data that I promised you, so let's go that way. Where there's a field of laminar fMRI, which there are quite a few people in Nijmegen who are also looking at this. The very first paper that I read where there were laminar responses was this one, the M paper, where they were projecting an M on the cortex of the subjects, and they used it to show that the definition of that letter gets worse as you go to the surface. So it's also a measure of how bad are the draining veins and what really, if we know that there is what the true pattern is below, how well can we read that out from the fMRI data. This is as a function of cortical depth, and there are definitely changes. So this is now something we can do, really. So if we can look at different depths, and we have from the electrophysiology, we know what layers of tissue are supposed to, say, deal with input, deal with output, then we can also design experiments in such a way that we can look at the input and the output of a particular piece of cortex separately, like Renzo Huber did here in his neuron paper. And the nice thing of this is that you can now ask different questions, because before we could just say, does this part of the motor cortex respond, yes or no? Is it maybe positive or a negative response? But now we can say, is there a change in the input or is there a change in the output to this particular piece of cortex? And it's really the step to sub-millimeter resolution that has made this possible. Of course, there's always other dimensions that we'd also like to explore. The first is that of the cortical columns. They're largest, but still very small in the visual areas. And that first experiment actually was a single slice where they were averaging a rather thick 2 or 3 millimeter slice. And now as the hardware gets better, we can see if we can improve on that. So and maybe combine information from layers and columns. So this is an experiment that we did in Amsterdam, where we looked at the ocular dominance columns, and then also as a function of cortical depth, so that we can ask, if we attend to one eye or the other, can we drive the changes in those well, two ROIs and for the different input fields. So if we move beyond the sensory maps, there's I already mentioned the numerosity maps. So if you think of it as a food problem, let's say, then 3 bananas is less than 6, and then we can construct maps for how we perceive these numerosities. And those are the modeling is done with a PRF-like design. They're pretty reliable, and also here we can look for short-term plasticity. So where if you present a numerosity range, so get people to look at, say, 1, 2, 3, 4, 5 bananas, and then you show them 20, then suddenly their maps will shift depending on what is a relevant quantity. And you can see here the higher ranges will sort of move over the slower one, the lower values on the right. In an experiment which was inspired by those early numerosity maps, we also looked at time mapping. And here the task is to judge durations. And it's the same thing as with the numerosity. We don't have a sense for this, but it's very important to our daily lives. And it's also something that everybody can do. We're actually pretty good at this. If you're catching a ball or playing tennis, this is an important skill, and people will be able to time these sort of sub-second durations amazingly well. So here, Domenico had, this was done in collaboration with Domenico Butti, and she had these ideas of which ROIs we should be seeing. And then if we look at the IPS, we actually get a nice rainbow-like progression, which for the longer and shorter durations. So why is it now then important that we do all this in single subjects? I think that for clinical applications, really what matters is the one patient. If you have, not all patients are all different, but also if you want to help someone, if you want to say the expectation of this treatment is like this for you, you only have an N is 1 ever. So I think it's important that we get to a place where if we want functional MRI to contribute clinically, we should be able to make these statements based on individual subject data. So the clinical applications that I did work on were following. They are then based also on these maps or mapping strategies that we did before. So with the tonotopic maps, you can do an attention modulation, but we can also look at what changes in say long-term damage for as occurs in tinnitus patients. So here are the frequency maps, and now we see that for these patients, there's an overrepresentation of the low frequencies, which is a bit surprising because they often hear a very high beep. So this is something that you wouldn't expect if you're, or that we didn't expect at least prior to the experiment, but it's very consistent. So we see always that these lower frequencies, they take up much more space in the cortical representations than they do in healthy controls. And in the somatotopic mapping, we were very lucky to find a very small group of patients which had lost a limb and then since had their nerves rerouted so that they could still feel what the non-existing limb. So if we look at the maps, of course we have them bilaterally. This one's only one-sided. And if you look at then the healthy controls, we see them as you would expect on both sides, and in a standard amputee group of patients, we see that they're completely unilateral. But if you then replace the input to that missing limb, we see that there's also that the representation of the hands is still there. So there's multiple fingers, and in the region where where you would expect also the representation of that limb. And again, the patient numbers here are so small. This really has to be done in individuals. In a similar vein, the use of 7T in epilepsy has become more widespread. And I think here there's a lot of work being done on the anatomical scanning, because you can see the lesion sites, see a lot of the malformations that cause the spiking that you're then interested in. But I think there could be a role also in the fMRI here. So we did a pilot experiment here where we also included EEG, and then you can deduct from the EEG trace what the bold correlates would be that they're looking for, and then you see that the clusters that you get for this the volunteer here, that they are much more defined, they're much more limited to the gray matter. So if you were going to operate this patient, you would then know better where you would have to go. So here it's also the high spatial resolution that's really important, and the higher bolt sensitivity, because it just makes it more feasible to do this in that one individual. And then as I was preparing the talk, I thought I should also look for things that are maybe not so what do you call it, current, a bit into the future. See where are we close to applying functional maps in a clinical context. And I think one of the most logical ones would be multiple sclerosis. And there's a lot of work being done at ultra high field at MS, because the lesions are small, they're much easier to identify when you're at higher field, higher spatial resolution, you get slightly more contrast. So if the lesion detection is improved, it's also easier to then link that to any symptoms that you do find, because you're better at finding them, so your explained variance is higher. So this is a figure from the recent paper from Madsen et al., where they did that. They counted lesions in the motor area, and then they matched that to their motor scores, and then you see that you can explain quite a bit of variance just by looking at the presence of the lesions, but you do have to be able to see them in the first place. Now, of course, if you're that's a link between symptoms and anatomy, but there could be a role there for functional MRI in the middle, where, especially if we're looking at things like networks and network integrity, we could see that better in individuals. If we have more bolt sensitivity, we could probably make a better estimation of how well is a particular network surviving over time, and how much of the variability in the network can we follow in a successive imaging session. This is something I would really be interested in seeing, but I don't think that this is quite happening just yet. Who knows? I already said maybe next time I come in person to give the talk, I can tell you some of this. And another one where I think fMRI could be clinically useful is in deep brain stimulation planning. So in the Spinoza Center, we scan a lot of, well, we don't. The neurosurgeons scan a lot of their patients which get deep brain stimulation for different diseases, but they're always looking for the anatomical target. So, which for Parkinson and essential tremor, it's the subthalamic nucleus, and then especially the motor sub-area of the STN. And this is actually a figure from a paper of theirs where they look at 1.5T, 3T, and then 7T. And if you're not terribly familiar with the STN, sorry, I just have to move the figures, then it's outlined here. And you have to be able to distinguish it from the SM, which is just below. So we're seeing two sort of little black smudges, and we're aiming specifically for the top one. And if we add a diffusion scan to that, then we can also subdivide it into the different subdivisions. And as these are mainly motor symptoms that they're aiming to treat, then you also have to aim the electrode for the motor compartment. And of course, it's helpful to have the anatomical information here, but it would also be good to back that up with functional information. And again, these are individual subjects, and there's there's a huge amount of variance in between how big the STN is and exactly where it is placed. So it's essential to have this information in a case-by-case basis. So I think it's very true to say that 7T is a game changer for neuroscience. I'm less sure about clinical neuroscience and clinical practice. Well, that's even a bit further into the future, but for neuroscience, definitely. So we have unprecedented spatial resolution, both functionally and structurally, and provided that we have the right questions to ask, we can really make a difference. It's a beautiful moment for Zoom to hang. So there's also the possibility now of running more subtle tasks, which, similar to the higher spatial resolution, means that you can ask questions that you cannot ask at lower fields. And you can do so in brain regions that are less easy to explore at more clinical field strengths. So with that, I have lots of people to thank for sharing slides and figures and generally helping out. And I'd like to thank you for your attention.
Amazing. Thank you so much. I got a million of questions, but I'm going to hold the floor for everyone else to get started. Are there questions either live here on Zoom? You can raise your hand and ask the question directly, or also over on YouTube. If you write your question, I'll ask them. First one up is Valentina.
Hi, thanks for your talk. It's been very educational for me, coming from cognitive neuroscience, and very interesting. So coming from cognitive neuroscience, I have a question on, is the 7 Tesla applicable to group level studies? And with this level of precision of the activation and resolution, how one can account for inter-individual variability at the group level?
So what the field is doing, I think now, is to define areas and then average those. So in the tonotopic maps, for example, you could look at all the Hessel-Jarvis, align those, and say these are all the low frequency areas, and I'm going to pool data then at that resolution. But I think what we really need is a better standardized space, let's say, because if you're moving it into the MNI space and it's 1 millimeter, then you lose the extra that you have. Having said that, the human connectome data, that retinotopic map where you go all over the brain, that is a group data set. I think there's even 180 subjects, and they did take a lot of care with the distortions corrections and so on. So it is possible to pool data over subjects, but you lose, of course, things like cortical depth. That's just not something that's easy to remain. Then you really have to work with, so you define an ROI and then define the cortical depths in an individual, and then pool all the different, so if you have eight different cortical depths, you can say, okay, I'm pooling those across my subjects, and then I average.
Thank you.
Thank you. I'm going to give everyone else a moment to be brave and raise their hand. Following the question on inter-individual variability, I was wondering if you see more variability on a 7T scan than you would see on a 3T or 1.5. So the idea being that if you have a more boxy acquisition, you blur over more of the variability, and when you actually have the 7T or even higher, you see more and more details. So I would expect a more. I
think so, but people are really quite different. So, for example, that auditory cortex measure, sometimes people have a split Hessel-Jarvis, and those are a centimeter apart. So that's a sort of variability that's very easy to see even on the 1.5T, I think. So yes, you would see more of it. I think you would also, like that example that I just gave, if you're looking at different cortical depths, you could say, okay, I'm always doing so many millimeters from the top. And then you see that people actually vary in how thick their cortices are, even across the ROI. So I think you have to always make a choice. So how do you deal with that variability? If you know that, say, younger people have thicker gray matter, then look at things as a percentage of cortical depth and not as millimeters from the top. So there's one of the other places where we see a lot of variability is in cerebellum. So we look at these high-res things, and then the folds are, but it's also small. It's difficult to pull that properly, and then the folding is actually different, and it gets worse when you look at patient data because the atrophy doesn't happen in the same way for everyone. Every slight variance of a disease is then slightly different. So it's it's a bit of a lame answer, but I don't know how much I would not see on a 1.5T. I've been looking at 70 images for so long. It's a good question.
Fair enough. Follow-up question for me before I give it back to our attendees is, you mentioned it's a game changer for neurosciences, but probably will take some more time until it's actually clinically relevant. And I was wondering, rough estimate, how many scanners out there are actually able to acquire 70 images at that quality at the moment?
There's roughly 100 installed now worldwide. There's an installed base of 100, and a fair percentage of that, but I'm not sure how many, are also getting CE labeling. So they would be a bit easier to apply to clinical data. So yeah, that's roughly the sort of number that you're looking at. It's doable. We can make it, but compared to the installed base of 3Ts, it's nothing. Right?
Of course. Ahmed has a question for you.
Yes, hi. Thanks for this very educational talk. My question is, I've been recently playing with 70 for a visual experiment, and the main, I think, challenge I faced is A, of course, distortion correction, but B is also sensitivity to physiological noise. So I think, of course, with higher SNR, you also get higher amplification of any interference coming from noise, not from the task itself. And I guess to get these really high resolution acquisitions, say 1 millimeter isotropic voxels and so on, you'd be using, at least what I was using now was 3D EPI rather than the standard 2D EPI that we get in typical 3 Tesla experiments, which I guess also amplifies the sensitivity in certain places, especially subcortically, to physiological noise. So my question to you is, are there any nice tricks out there to use for still achieving large coverage with high resolution, but reducing sensitivity to physiological noise other than post hoc correction of this data, say something at the acquisition level?
Well, so I think it's the when you say whole brain and submillimeter and night sensitivity, that's a very, like, that's already a very difficult combination of factors.
Yeah, I know.
Given that I don't know, if
really
you happen to be based in Nijmegen, you could try a multi-echo approach. You could see if a shorter echo time would give you information about the non-bold factors, and then look at the. I haven't tried that, and I'm not sure how easy it would be, but if there is anywhere that you could make it work.
I think the main interest here is to sort of map a subcortical structure and a cortical structure simultaneously. So something like LGN and visual cortex in one go, rather than having to do an LGN targeted acquisition, because then
then you wouldn't really need it to. In that case, then I would always advise you to take a slab which goes through the LGN and the visual cortex, but not insist on the whole brain.
Yeah, it wouldn't be whole brain in this case, but it still has to have some sort of decent coverage to get several cortical areas involved. But
and what also helps, that might sound silly, but is very basic, right? Make your subject so comfortable that they don't move when they breathe. So
Yeah.
if they're very, right, if you see that there's respiration related movement, then I think that is something you can actually prevent from happening. I've also done experiments at maybe slightly higher spatial resolution, especially if you're looking at the LGN anyway, which is very small. Then you can say, oh, I have slightly smaller voxels, not so much. I'm not so bothered by the physiological noise then. But you'd have to, it would depend on how good your image quality was, whether you could actually get away with that.
Right. And then in terms of analysis, you were mentioning earlier that we're sort of always used to this standard pipeline and so on when we look at the data. If I'm looking at BOLD here, I'm not looking at CBF and so on, or blood volume, sorry. But even in that context, do you have any tips about some other approach to use in analyzing the data? Now that, say, I've pre-processed and done everything, and now I'd like to just run my, say, GLM or something like that on the data. Anything I should keep in mind within this context for 70 in high resolution?
No, I think I'd have advices about the pre-processing in the interview. No, not smoothing, or as little as possible. In a GLM, the BOLD responses are not intrinsically different. So if you have a stimulus model and you can evolve with a standard HRF, that should still be a good model. You could, I think, for the LGN, and I'm not sure because I never looked, but you might expect that the HRF is actually a bit differently shaped.
Right.
So I would always include things like a temporal derivative, maybe dispersion derivative, and check very carefully that the responses match your model.
Yeah.
Because they're, because they might not. That's subcortically, that can be very difficult.
Right. Well, thank you. Thank you very much.
Thanks, Ahmed, for the question. A couple of more questions, if I may. So you've shown a lot of clinical examples, which is fantastic, but I couldn't help but think if it was applied to language and brain tumors, for example.
Whether I know that it has been.
Because I'm not aware of it, but then.
No, so tumors are even more difficult to pool across subjects, right? Because they're, no, that's a good question, because I think there should be like ways of, doing better on your pre-surgical planning, if that's what you're.
That's exactly where I'm going, yes.
Yeah. Yes, no, good idea.
There we go, common project. The other question I had for you is that you just mentioned in your answer to Ahmed that you need to make your participants comfortable to reduce the artifacts. But I was wondering if the comfort for the participant or the patient in the scanner is different in a 7T compared to a 3T. Like, do they feel the increase in the field strength? Or?
Yeah, so we scan a lot of people that come from outside, right? Like our normal controls. And what we hear a lot are, so we get basically two types. You get people that have never been scanned and they think, oh, an MRI, this would be fun. And they show up, and then we tell them that they have to remove all metal, and they get in a magnet, and they come out, and they happily go home, right? And they never even, we don't really even need to tell them that it was a 7T. And then there are people that, especially when they work on it, so people like you who think, oh, this would be cool, let's go do a 7T study. And they're very alert and also aware, and they know, right? And then, and also myself, then you feel that you get more dizzy, that the metal taste that you have in your mouth is stronger than you do on that, than I do on the 3T. But it's, and it's also dependent, I think, on the stress levels of the subject. So when we get people that are very nervous, then they experience more of this turning sensation. But yeah.
That is actually how I experienced it.
There you go.
I was in there and I was like, oh, this is this sequence, this is this sequence. I recognized the sounds. But to me, it felt different. And a normal 3T scan.
Yeah, it does. It would do. I mean, yeah.
Valentina has another question for you.
Yes, I was wondering, can 7 Tesla technology help us to improve our knowledge on how the brain functions? And in particular, can we explore whether there is activation in the white matter, not only in the gray matter?
I was going to say that whether it can help you explore how the brain functions depends on how good a neuroscientist you have asking questions. But yes, so there is, I forgot who it is, lab in this day, John Gore is looking at white matter responses in, I think, on a 3T, but so the same applies, that they are bold responses. And so you would expect that with increased sensitivity to susceptibility, you would see larger responses. I've never tried, so they do these experiments with where you expect a lot of cross-callosal communication. And then you can, because then you have a nice big white bundle that you know what it's doing also. And I've never tried that, so I don't recall that I've seen consistent white matter responses ever. But yeah, I think if you were going to do it, then certainly would help there. Plus, you wouldn't have half of these problems with the normalization, because the white matter is like nice, big, easy to pull across subjects. So maybe that is something that you could actually apply a standard pipeline to, and it would just survive quite well.
Maybe also with different, like instead of the bold signal, the CBEV.
Yes, the CBV. So the CBV signal is anyway quite small.
Okay.
And the CBV percentage, there's less blood in the white matter than there is in the less well perfused, and the cerebral blood volume is also lower. So
okay, that's it.
I think looking at white matter responses with CBV would be very difficult.
All right, thanks.
Lovely. That's all the questions I can see at the moment. Thank you so much for this fascinating talk and for your time. Little recap before I let you go to the beach is that this was the last talk for the first half of this year. We're taking a summer break and we'll be back in September. That is the 29th of September, Thursday again at 4 o'clock, with a talk on the involvement of the cerebellum in motor and emotional learning by Dagmar Timman. So do join us for that in September, but we put the notifications on social media so you can't miss it. With that, I wish you a wonderful summer break and see you back soon after the summer. And for everyone in this session at the moment, thanks for being here. Thanks for joining us over on YouTube, and I hope you can all go outside and enjoy the sunny day now and go to the beach. Thank you so much for being here. Bye.